Alright, so for drawing biomolecules, you need to first know the elements that will be in most of the biomolecules that we are drawing. So, we have carbon, hydrogen, oxygen, and nitrogen are the four that you really need to know. And then you need to know the number of bonds that they can make. So carbon can make 4 bonds, hydrogen can make 1 bond, oxygen can make 2, and nitrogen can make 3. We discussed all of this in class. You just need to know these things. It will help you later on. Right. And then what's important to know is the different types of functional groups. These are things that connect to a carbon or some other element in your molecule. When we draw the molecules, you are first going to draw the carbon backbone and then add in your functional groups where appropriate. It'll make sense later on when we do it, but it's important to know these. So the hydroxyl group is when you have usually a carbon connected to an OH, an oxygen, and a hydrogen. You could also show it like this if you wanted to show all of the bonds there. Either is acceptable. It is called a hydroxyl because hydrogen is in it and oxygen is in it. Hydroxyl. We then have the carbonyl group. This is a carbon connected to double bond oxygen. That leaves us with 2 other bonds that this carbon can make later on when you fill in the rest of it. We then have the carboxyl group. This one's kind of like a mix of the 2 that we've already done, as you can tell by the name. Hydroxyl, carbonyl. This one's the carboxyl. And it looks like it's a mix. You have this carbon double bonded to the O like the carbonyl that we just did. And then another bond to an OH. Again, you could also draw it showing all the bonds where you have the O and then the H like that. Both of these are okay. In the carboxyl, this leaves this carbon able to make 1 more bond because it already has 1, 2, 3. So you want to make 1 more bond with something. And we will later on when we get to filling in. The last functional group is the amino group. This is going to be only found really in your amino acids, which, reminder, make up proteins. So you're going to have a nitrogen attached to 2 hydrogens as your amino group, which makes sense because amino acids make up proteins. Proteins are the only ones that have nitrogen, so it makes sense. Nitrogen likes to have 3 bonds. This group leaves it so that nitrogen can bind to one other thing, which is usually the rest of your amino acid. Okay, so those are our functional groups. Now we are going to move on to the actual molecular golf. This is the worksheet, so hopefully you will follow along and draw what I do. I recommend that you use different colors of pens or pencils just so you can like really make sure you know the difference between the functional groups and where they go, because that is important. So the first one on the sheet is ribose. This has a chemical formula of C5H10O5, and you're going to see with carbohydrates, this is a carbohydrate because it ends in ose, so it's a sugar. Carbohydrates tend to have this chemical formula structure where you have 1 carbon to every 2 hydrogens and then 1 oxygen to every 2 hydrogens. So it'll always kind of be a ratio like that, if that makes sense. If it doesn't, tell me. I'll explain it in better words later on. All right, so first you're going to draw your carbon backbone. This is usually like the ring structure that we talk about when we're drawing carbohydrates at least. So for ribose, this is a 5 carbon sugar. So you are going to be making a pentagon. So for this, we have 1, 2, 3, 4 carbons, like this, in the actual backbone. But then at the very tip top, there is an oxygen. And this happens a lot with with carbohydrates. You will have an oxygen somewhere in the ring just because it makes it close, because oxygen always bonds to two things. So this oxygen is happy where it is, it's chill. It does not need any more bonds. These carbons need more bonds though, because most of them only have 2. So this carbon right here needs 2 more bonds. This one needs 2 more. This one needs 2 more, and this one needs 2 more. They all need 2 more bonds. The 5th carbon, because we only have 1, 2, 3, 4 carbons here, comes from being attached to this one. So in the upper left, you're going to have a C as well. This carbon is going to have a hydroxyl group off of it. So you can draw that right now. And actually I'm going to do the hydroxyls in a different color. And I suggest you do too. So I'm going to do pink, because I like pink. So I'm going to draw the hydroxyls. Where they go does matter, because if you put it in a different place, it will make it a different molecule, so it would make it a different sugar. So you need to make sure that you put it in the right positioning. So off of this one, we do have an OH. Off of this carbon, on the top is an OH, or a hydroxyl group. Off of this one, we would draw it over here, hydroxyl group right there, another hydroxyl group, and that is it. So that's it for our hydroxyl groups. But you might be thinking, this carbon right here, right, only has 1, 2, 3 bonds. Where does the other bond come from? Your last step is going to be to fill in any missing bonds on carbon with hydrogens. So you would do a hydrogen to fill that in. 1, 2, 3, another hydrogen. 1, 2, 3, another hydrogen. You can draw it like that. You could also draw it on the side. Doesn't really matter much. This carbon, 1, 2, we would add 2 hydrogens to this one. Okay? So this is our finished ribose. I'm going to draw it at the bottom again, just to make it a little neater. So we would have O at the very top, carbon, 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 and a carbon there, 4 carbons in the ring. Then I would add another carbon up here, and if you wanted to, you could even do CH2OH up here if you don't have enough room, like on the paper or in the box. This is also an acceptable way to do it. And then I would fill in my hydroxyl groups. I know it goes on the top of this carbon. I know it goes bottom over here, over like this. And then I fill in any missing bonds of my carbons with hydrogens. So hydrogen, 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 and hydrogen. So this is my ribose molecule. And if you count every single carbon, hydrogen, and oxygen, you will find that there are 5 carbons, 10 hydrogens, and 5 oxygens. And that's the same. So if you ever want to double check your work, you want to go through and count the elements that are in the molecule just to make sure that it matches the formula that you know. Okay, so that's ribose. We're going to move on now below that one in the box to alpha D glucose. Oops. This is, you'll notice there's 2 glucoses. That is because based on where the hydroxyl is placed in some of them, that will determine whether it is beta or alpha glucose. Okay, so that's why it's important to know the functional groups and to know where they go. Otherwise, you will get the two mixed up. So alpha D glucose, we are going to start with our carbon backbone. All right, we are going to start by knowing that carbon, there are 6 carbons in glucose, 12 hydrogens, and 6 oxygens. Again, following that pattern of 6, 12, 6, there's always going to be double the amount of hydrogen as there is carbon and oxygen in a carbohydrate. That's just usually how it is. So now we are going to start with our carbon backbone. Again, that's going to be our ring. So this one is a 6 carbon sugar, so it is not going to be a pentagon, it is going to be a hexagon. So we're going to start by going with that. It starts at the top with a C connected to an O, and then you do 1, 2, 3, 4, 2, 3, 4, 5 sugars, carbons. Alright, so we are going to start with our carbon backbone. For glucose, there will be 1, 2, 3, 4, 5 carbons in the backbone, and then 1 again is going to be attached to one of those carbons. So you start off with a C connected to an O, carbon and an oxygen, and then you just fill in 1, 2, 3, 4, 5. That's kind of ugly, but you get the point. I'll do it neater later. And then up attached to this one, you're going to have that similar thing that we did with the ribose, where you have a carbon. You can write it like this, CH2OH if you don't have enough room. Or you can do the bonds, where you have like OH, H, H. Okay? For alpha D glucose, the important difference is that on this carbon right here, the OH is at the bottom and the hydrogen is up top. When you compare the alpha and the beta after looking at it and going and drawing all of them, it's good to like circle the difference. But I'm just letting you know that this is the difference right now. I'm going to redo this so that my functional groups are all in the same color. So I'm going to do pink again, and I'm going to put my hydroxyl group there because I know it goes there. I have a hydroxyl group that goes down here. For the next carbon, it's inside the ring. For this one, it is on the bottom. So those are our hydroxyl groups. I'm going to circle the one that is different from beta D glucose for now. Okay, and then again, 1, 2, 3. The carbons are not happy. They need a final bond. You fill in with hydrogens wherever there is not a bond. Okay, so this would be our finished alpha D glucose. If you again go and count, you would have 6 carbons, 12 hydrogens, and 6 oxygens. You would know that you did this correctly. Good job. All right. I'm going to draw it a little neater down here, just so you have a better understanding of how it should look. Like that. Sorry about the Apple Pencil clicking. I know it's probably annoying. But yeah, so that is alpha D-glucose. The only difference between this one and the one we're going to do next is this one. So in the next one, it is going to be flipped, and this will be right here where this hydrogen is. So beta D-glucose, it has the same chemical formula, C6H12O6. It is structurally different though. So when we draw it, we're going to go with the same steps as we did with alpha D-glucose. We'll do C connected to an O, C, C. Those are our carbons in the ring. We have our CH2O, H right there. The difference is that right here on this carbon, my functional group is going to go there. So my hydroxyl group goes up top there. All the other hydroxyl groups are the same as the other glucose, so that makes it kind of easy, or a little easier at least. So again, that's the only difference. Then you go in and you fill in any carbons or other elements that are not happy with the hydrogens that are missing. And that is your beta D-glucose. I think I did this one pretty neat on the first try, so I am not going to redo it. You get it. All right, so I would make sure that you circle these on both the alpha and the beta, just so that you remember that is what's making them different from each other. Okay, so those are the carbohydrates that you need to know how to draw. I'm going to move on to the generalized amino acid in the top right corner. This is a lot easier. The carbon backbone is not going to be a ring anymore. It is going to be just one carbon for the amino. So you have one carbon as your backbone. Carbon makes 4 bonds. You are then going to have a amino group because it is an amino acid. So you have a nitrogen connected to that carbon and then 2 hydrogens. And then you are going to have a carboxyl group, which is going to be another carbon with an O and an OH. Let me do this in a different color as well. Okay, so you have your 2 functional groups here. This is the amino group. This is your carboxyl. If it helps you to do this on your paper as well, feel free. You will notice that the carbon has 2 unfilled bonds. Well, from what we talked about in class, the amino acid is going to be a generalized amino acid because there are 20 different ones that we could do, but you don't need to know how to draw the different 20. So we're going to fill in with a variable. So you can do an R with a little box. That R is your variable. That could be any combination of elements that will make it a different amino acid, but all amino acids have the rest, that carbon, that carboxyl, that amino group. Every amino acid has that function or that structure. So that's not going to change. The only thing that's going to change is the R group. That's your variable. You don't need to know it. You just need to draw an R. Okay? Now we have one more bond. We're going to fill that in with a hydrogen. There's your amino acid. I'll draw it a little more neatly down here. You have your carbon. Then you have your amino group. And then you're going to have your carboxyl group. R group, your variable, and then a hydrogen at the top. So again, we're following the same steps of carbon backbone, functional groups, then filling in with hydrogens wherever bonds are not happy. So you would go around and you would be like, nitrogen has 1, 2, 3 bonds, it's good. Carbon, 1, 2, 3, 4, that one's good. 1, 2, 3, 4, that one's good. Every line is a bond that you're drawing. So even if it's a double bond, that counts as 2 because it's double bonded. All right, last one, yay, fatty acid. Oh my god, I cannot write. I'm so sorry. Fatty acid, also known as a lipid. Okay. So this is, again, generalized because fatty acids can be different lengths. So we are going to start with our carbon backbone, and I'll explain that a little more later. So when we talk about lipids and we looked at lipids, you noticed that they have long chains on their ends that have only carbon and hydrogen. So our carbon backbone is going to be carbon connected to 2 hydrogens, and then we're going to go and put it into brackets, and then write N. That N equals, again, a variable. Depending on the fatty acid that we're looking at, the lipid that we're looking at, it could have 2 copies of this CH2. It could have 100 CH2s binded together. It really depends on what we're looking at. It then is going to have a CH3 at the end, just to make sure that it gets capped off, and then a carboxyl group on the other side. Okay, so I'm going to draw this assuming that N is equal to 2, just so that you can see what it would look like fully, but this is what you would have to draw, okay, on like an exam. So the example that I'm talking about here is, let's say we had CH2 repeating twice. So we would have 2 carbons bonded to each other. They would each have 2 hydrogens on them. Then we would have it connected to a C with 3 H's. This carbon and this carbon have all of their bonds filled. They have 4. They're happy. This one only has 3. That's where the functional group comes in. OH, O. Okay? So that's what it would look like if N was equal to 2. If N was equal to 100, it would be way longer, and I'm not drawing all that. So hopefully you understand. If you don't, talk to me on Monday or whenever, and I'll go over it. But again, I'm going to draw the one that you would need to draw in your box so that you know your carbon backbone is CH2 in brackets with an N connected to a CH3 with the functional group of a carboxyl. So that is your generalized fatty acid. Right, so these are the 5 biomolecules that you could be asked to draw on a test or on the IB exam even next year. You could also be asked to identify one, so I could give you a picture of a glucose, and I could ask you, which type of glucose is this? And you should know it based off of the picture. So really go through, rewatch this video if it helps. Keep drawing them. Draw it in your notebook. Draw it again in your notebook. Draw it multiple times to practice drawing them. That way it really sticks in your head. And then follow the steps, because I think the steps are really helpful. Maybe not so much for this one, but for the other 4 they help. Okay. With that, you are done with your homework. Congratulations.